What that is, if we zoom in there, are a bunch of actin filaments that are cross-linked with each other a little bit loosely, and also anchored onto the attachment sites on the plasma membrane. So if you grab any two actin filaments and pull on them, what you're going to be doing is you're going to be bringing together different points of the plasma membrane. So what happens at the back of the cell is that the cell polymerizes regionally short filaments of myosin II, short myosin thick filaments, and by their interaction with those actin filaments in the cortex, they are going to produce contraction. So follow this myosin here. This myosin's heads are going to be pulling on this actin filament. These myosin heads are going to be pulling on that actin filament. And in doing so, you're bringing the top and the bottom together. So that's what's going to cause contraction of the back of the cell. Yeah?
The myosin II fixed in place.
The myosin II?
Fixed in place.
No, it's floating around. It's all relative. So if I have ropes around it here, and I'm just floating around, I can grab any two, and if I pull on them, I'm going to be bringing those two walls together. All right? So that's how non-muscle cells move using essentially the same machinery, but not as well organized as we find in the muscle. Questions? Any lingering questions before we move to intermediate filaments? Yeah.
Do those myosin molecules just randomly grab two actin molecules and pull?
They will grab whichever actin filament they can find. Yeah. They don't have any way of discerning where the actin filament is coming from. All right. So those are actin filaments. Intermediate filaments are called intermediate filaments because they are in relative in size. They are in between actin filaments, which are very thin, and microtubules, which are quite thick. So they are in the middle in size and thickness. Intermediate filaments also form vast arrays of filaments within cells, as you can see in this micrograph. These are micrographs of skin cells growing in culture. And in a very light blue, we have a highlight of their plasma membrane with attachment sites in there. Nucleus, basically devoid of intermediate filaments, and lots of filaments all around the cell. What you will notice in this picture is that many of the intermediate filaments from one cell end up at the plasma membrane at the same spot that intermediate filaments from the adjacent cell. So what we're going to see through a little bit of today, and then again when we come back to cell attachments, is that cells are able to form networks not just of one cell, but of many cells in a tissue to have this mechanical strength together, so which is particularly true of skin cells. All right? So intermediate filaments are really good at holding shape and giving the cell mechanical strength against shearing forces. So when you put intermediate filaments in a cell, they're going to be strong individually, and then when you connect them to the intermediate filaments of the neighboring cells, now you have a collective of cells that is mechanically very strong. We see that in many tissues around the body, but it's particularly true and obvious in the skin, which is subject to a lot of friction. So let's talk a little bit about how, a little bit more about it. So intermediate filaments, and again we're going to come and visit this after spring break. We're going to see that they are attached to integral membrane proteins on the plasma membrane that can stick two cells together, or that bind to integral membrane proteins that bind the cell to the extracellular matrix tissue, so that they can form a mechanical, a nice tissue layer. What are these filaments made of? They are made of a protein. What are the subunits? The subunits is a protein, soluble, these are all soluble proteins, cytoplasmic proteins, therefore no other kinds of signals in them, right? That very different from actin and from microtubules, which are globular proteins. Intermediate filament proteins are very elongated proteins. And something that is very elongated, a protein that is very elongated, we call it fibrous, because it forms kind of like a fiber. Right? So, and with actin, we just talked about the actin monomer, G-actin, single protein, globular protein, all by itself floating around. In the case of intermediate filaments, we have not one, not two, but four polypeptides that come together to form the subunit of intermediate filaments. So here's a monomer that was produced by a ribosome. You can see it's a very long protein, a tiny globular domain at the N and C terminus. The rest is a very long alpha helix. That alpha helix grabs onto other copy to form a coil, like we talked about the myosin tails. So they form a long, strong coil. These two guys are totally like a twisty, totally bound together by hydrophobic interactions. But then, and this is the tricky part, you take one dimer and you take a second dimer and you let them bind together, but not in the parallel position, but in the anti-parallel position, and not perfectly registered, but they are bound together with a stagger. So here's one dimer coming from here to here. There's a stagger there, an empty portion, and the opposite dimer running in the opposite direction. This interaction is also caused by hydrophobic interactions, lots of hydrogen bonds in there, and it's a very, very tight binding. So the tetramer, once it's synthesized, hangs on together for a very, very long time, basically for the lifetime of this protein. So the tetramer, once it's formed, you can think of it as a single unit. And that's the subunit, the soluble subunit that can be added or removed from an intermediate filament. So not the dimer. This is a common confusion. It's not that single dimer that comes off or assembles onto a filament. It's the tetramer. The tetramer is a functional subunit of intermediate filaments. Now, you will notice immediately that this subunit here, which is a functional subunit of intermediate filaments, is completely symmetrical. You could flip this 180 degrees, it will look identical. Therefore, this subunit does not have polarity, unlike actin, unlike microtubules. Perfectly symmetrical, because it's formed by two subunits going in opposite directions. So that means that when you assemble the intermediate filament, the intermediate filament as a whole will look identical if you flip it 180 degrees. Means no polarity. Intermediate filaments are not polar filaments. They are symmetrical filaments. One more strange thing about intermediate filaments compared to actin. With actin, we said that actin subunits could only be added at the ends, never in the middle. You could not add or lose subunits from the middle. That's not true for intermediate filaments. With intermediate filaments, you can add them at the ends, you can add them in the middle, you can remove them from the middle as well. And that's what's illustrated out here. In this figure, to make it, I just flip it 90 degrees so it fits better. But just look at here. This subunit, which is this cylinder, which represents a tetramer, can come off from there, leaving an empty spot that can be filled in by a brand new subunit. So there can be a lot of remodeling of intermediate filaments in the cell, simply by changing subunits in the middle of the filament. This assembly and disassembly of subunits, therefore, is not going to be as tightly controlled by critical concentration and all of those things that we talked about with actin. Instead, it's going to be very tightly regulated by a series of kinases and phosphatases that are going to change the phosphorylation state of the molecules and therefore their ability to bind together or not. So phosphorylation is going to cause the disassembly of the intermediate filaments. If you remove the phosphate with phosphatases, they will be able to rebind together. We are going to talk about one universal example of intermediate filaments that is found inside the nucleus. I already mentioned that there are filaments inside the nuclear envelope in the inner side called the nuclear lamina that gives shape to the nucleus. That nuclear lamina is composed of a unique kind of intermediate filaments found in all eukaryotic cells. And if you phosphorylate them, they disassemble. If you remove the phosphates, now they can reassemble. And we'll talk a lot about that when we talk about cell cycle and mitosis. Questions so far? Now the fact that you are putting together molecules that are twisted around each other, long proteins, and then you put two of those together that are bound like that, and then you make rows. Oh, I forgot to say, they make rows just like with actin. A single row is called a protofilament. I can read lips. So intermediate filaments have 8 protofilaments, unlike actin, which has 2. And the protofilaments are rows and rows of those long tetramers. So essentially what you're putting is long little ropes on top of long little ropes, making even longer ropes. So essentially the intermediate filament behaves like a rope in that it is flexible, but it's also mechanically very strong. So it has all the, here the rectangles represent the tetramers, and the tetramers are going to have lots and lots of interactions, side-to-side interactions with other tetramers in adjacent protofilaments. So you can twist this back and forth, and this intermediate filament will be able to withstand that friction, that torsion, that mechanical stress. That is not at all what you would see with actin filament, which is very thin and just has 2 protofilaments. You twist it, you can easily break it apart very quickly. It's also true of microtubules. Microtubules are very brittle in that if you put any kind of torque or mechanical stress on them, they break right away. But not intermediate filaments. Intermediate filaments will withstand a lot of mechanical shear. And that is why cells use them to hold together mechanically, to hold their mechanical stress against shear. With actin, I talked about just the actin. I didn't really mention, but you should know that there's many variants of actin in different tissues. So the heart expresses a particular variant, cardiac actin. Big muscles express striated muscle actin. Non-muscle cells express non-muscle actins. So there's a bunch of variety, but they are almost virtually identical. The diversity of intermediate filaments is even greater.